Hi all, I am Nucleic. Welcome back to our educational series on molecular biology. In this video, we will delve into the intricate process by which proteins are synthesized within living cells. Join us on this exciting journey as we explore the fascinating world of protein translation. Proteins are fundamental building blocks of life, carrying out crucial roles in various cellular processes. They are synthesized through translation, a complex process that converts the genetic information contained within mRNA molecules into functional protein chains. Let's start by understanding the key players in protein synthesis: messenger RNA, or mRNA, transfer RNA, or tRNA, ribosomal RNA, or rRNA. The process of protein translation begins with the synthesis of mRNA molecules through a process called transcription. During transcription, the DNA sequence that contains the genetic code for a specific protein is transcribed into mRNA. This single-stranded molecule acts as a messenger, carrying the genetic information to the site of protein synthesis. Now, let's talk about transfer RNA, or tRNA. This remarkable molecule acts as a carrier, shuttling amino acids to the ribosomes, the cellular machinery responsible for protein synthesis. Each tRNA molecule binds to a specific amino acid, and its anticodon recognizes and pairs with the corresponding codon on the mRNA molecule. Ribosomal RNA, or rRNA, forms an integral part of ribosomes. These complexes serve as the workbenches for protein synthesis. Ribosomes are composed of two subunits known as the large subunit and the small subunit. Both subunits are made up of a combination of proteins and ribosomal RNA molecules. The large subunit is responsible for catalyzing the formation of peptide bonds during protein synthesis, while the small subunit helps in reading the genetic information stored in messenger RNA. Genetic code and codons and anticodons. The genetic code consists of a series of three-letter combinations called codons. Each codon corresponds to a specific amino acid. tRNA molecules possess complementary sequences called anticodons, which bind to the codons on mRNA during translation. This precise matching ensures the correct assembly of amino acids into the growing protein chain. Wobble base pairing. Interestingly, some tRNA molecules can pair with more than one codon due to a phenomenon known as wobble base pairing. Pairing of the tRNA anticodon with the mRNA codon proceeds from the 5' end of the codon. Once the first two positions are paired, exact base pairing of the third position is less critical. The third base of the anticodon can typically pair with either member of the purine or pyrimidine pair in the codon as appropriate. It wobbles. In this example, the double ring G can pair with either a single ring U or C. Aminoacyl tRNA synthetase and tRNA charging. Before delivering amino acids to the ribosome, tRNA molecules must be charged with their respective amino acids. This process is mediated by a group of enzymes called aminoacyl tRNA synthetases, which ensure the correct pairing between tRNA and its specific amino acid. A, P, and E sites. Within the ribosome, there are three distinct sites: the A site, the P site, and the E site. The A site binds to incoming aminoacyl tRNA. The P site holds the growing polypeptide chain. The E site is where discharged tRNA molecules exit the ribosome. Initiation, elongation, and termination. The process of protein synthesis consists of three main phases: initiation, elongation, and termination. During initiation, the ribosome assembles on the mRNA, along with initiation factors and a special tRNA carrying the start codon. Now the first tRNA molecule in the P site. Next, tRNA molecule attaches to the complementary codon at the A site, and a peptide bond is formed between amino acids. The ribosome translocates three bases at a time, moving the tRNA molecule in the P site to the E site, and the tRNA molecule in the A site to the P site. The tRNA molecule in the E site will be ejected and a new tRNA molecule will bind to the A site, creating a new peptide bond in the process. Elongation continues the sequential addition of amino acids to the growing polypeptide chain. The ribosome's peptidyl transferase activity catalyzes the formation of peptide bonds between adjacent amino acids. Then, the cycle repeats, with translocation of the ribosome and the tRNA molecule until the full length of the polypeptide is made. Termination occurs when a stop codon is reaches, and the final polypeptide is released. Start and stop codons. The start codon, a UG, initiates translation and also codes for the amino acid methionine. Stop codons, such as UAA, UAG, and UGA, signal the end of protein synthesis and do not code for any amino acids. Molecular chaperones. Throughout the entire process of protein synthesis, molecular chaperones assist in correctly folding newly synthesized proteins. These chaperones prevent misfolding and aggregation, ensuring that proteins adopt their functional three-dimensional structures. And there you have it, a glimpse into the incredible process of protein synthesis. Hopefully, this video has shed some light on the intricate mechanisms that underlie the formation of proteins, the building blocks of life. If you found this video informative, don't forget to check out our previous video on transcription to further deepen your understanding of gene expression.